leading to the explanation about different larval forms and their significance. And in my previous class, I have explained in detail about the water vascular system, the various components of water vascular system. Now, regarding the other systems the hemal system and the reproductive system, they are not included in your syllabus. But time to time, we do refer to those systems while explaining uh, a particular topic. So I will be visiting those systems during the course of this class. So we know that these sea stars most of them are having one sex in one organism. They are not hermaphrodite, so they are unisexual, and they don't have both set of gonads in single one usually, but in some there are both gonads present, both male and female structures are present in single organism in some of the sea stars. While some sea stars or some echinoderms start as one sex and then they transform into the other sex during the course of development. How come people are still joining? I don't know. I have just told you that you should join by 10. So usually the sex ratio of male and female is 1:1 in the temperate coast sea stars and in echinoderms in general. So it is not that one particular sex outnumber the others. It is that both sexes are there in 1:1 ratio. These gonads, they mature through late autumn and winter, and spawning is when there is a relatively warmer temperatures and abundant seafood is available for the larvae, because larvae are feeding stages. So spawning is egg laying, and that is usually in the spring, coincidental with the seasonal appearance of warmer seawater temperatures, and there is abundant phytoplankton food is there for the larvae. So these larvae are like free swimming larvae, feeding larvae, and mostly there are larval forms present. But in some cases, the development is direct, that there is no larval forms, right? But in most cases, there are larval forms. So that way we say that development is indirect. So this is about the sea star, how they are, at what time they mature. Now, regarding the presence of gonads, so here you see these gonads. The gonads are located in this position on the oral side. On the oral side, here you see these gonads. So this is a TS, and here you see the bunch of these germinating epithelium is there, right? But if you look at the whole organism, where you remove the upper part of the skin here, and it is dissected, only one arm is dissected here, and the upper skin is removed. So you see they are present like this, starting from the base and reaching up to the tip part, almost to the tip part. Depending on their maturity stage, they are developed that way. The degree of development is directly correlated with the stage of maturity. And here you see that through this gonoduct, they are released to outside. That is, the gametes are released to the environment. Here, in this case, aquatic environment, in the seawater, these gametes are released. From male gametes are released, and the ova are released. So this is the arrangement. Now there is one theory that says that gametes are not formed in these structures. They are just stored in these structures. Rather, the gametes are formed here in this axial gland, here in the axial gland. And from here, they are transferred to these structures. They are transferred and they mature here, and then they are released from here. So this is just a opinion of a group of scientists. Some say that these gametes are formed here within this, while some say that gametes are formed in this axial organ here, and they are transferred from here to the gonads. So this is briefly about this. Now, because of the low parental investment in each egg, a female may have high fecundity, perhaps numbering in the hundreds of millions of eggs. Like the female lays about 1.25 million eggs per hour and about 200 million in a season. So you can see that there is an enormous number of eggs, as well as sperms are many times more than the eggs. So a huge number of gametes are released from males and females. These eggs are shed freely in water. Likewise, sperms are shed freely in water. There is external fertilization, and usually there is indirect development that includes the larval stages. So we know that how fertilization occurs when sperm meets an egg, it penetrates it, leaving its flagellum outside. Then fusion of both nuclei occur, and that leads to the formation of zygote. That leads to the restoration of 2n number. Both gametes are haploid. The zygote forms a fertilization membrane around it to prevent polyspermy. Then further development occurs. We know all those stages of development. We'll go one by one, go through one by one. The developmental stages from a single cell through early gastrula to the larva, and then maturation of larva, and then formation of adult, then growth of adult, like juvenile, first formation of juvenile, and then growth of juvenile to the mature adults. So the ova is an unfertilized egg. Nucleus and nucleoli are visible if you look through this under microscope, and it contains a small amount of yolk. So we call such type of eggs as microlecithal egg. So we see that there is not much yolk deposition. So there is very little investment in terms of matter and energy from the parent. So that is why the huge number of these eggs are produced. Then we know that sperms are produced in the male gonads, and they are released. They are motile, and they have a flagellum. They have that typical sperm structure. They penetrate the ovum, and the part of a sperm is left behind, and the nuclei is carried in there, and then the fusion of both genetic material occurs, and a zygote is formed. Zygote is a fertilized egg. It is a diploid. The nucleus is not visible here. We are talking about the slide here. That nucleus is not visible in the slide. Then cleavage division follows. We know that the cleavage is holoblastic. It's a mitotic cell division, total division, equal blastomeres are formed, not the micro and mega as you see in case of some higher animals. Then the plane is radial and it is indeterminate. That is, the blastomeres do not have fixed fate. Then once the division starts, a single cell it divides into 2, and 2 into 4, and 4 into 8, and 8 into 32, 8 into 16, and 32, and so on. So more and more number is added after every division. So initially it is just a 2-cell stage. Then it becomes a solid ball of cells, which we call as morula, which is a compact cluster of cells. Then the cavity developed that we call as blastula. So if you look at this picture here, we see this is a haploid gamete here having nucleus and nucleolus. Then after fertilization, diploid cell is formed, and then it is dividing. Here you see early stage, early cleavage, 2-cell and 4-cell, and then a ball of cells here, solid ball of cells, which is called as morula. And then the blastula and gastrula, the other stages, they come one after the other. First, there is cavity develop, that is called as blastocele, and then invagination occurs here, and that is the early gastrula. We will be studying these stages in detail in some of the next PPT slides. Here, this is under microscope, the real picture of various stages, 2 cell, 4 cell, 8, and then here kind of morula. You see the size of cell goes on decreasing as more number is added, bigger cells here, and the smaller cells you see in this as more number is added. Then here the early gastrula, late gastrula, and then various larval stages are shown in this picture. So we were talking about various stages in this, that is ovum, after fertilization, zygote forms, then 2 cell stage, 4 cell stage is and leading to the morula. Then morula is a solid ball of cells that transforms into blastula, which is a hollow ball of cells. And early, middle, and late stages are determined by cell size, which gets smaller with each round of mitosis. Now, this blastula is formed on the second day of cleavage. That means all these stages pass in quick succession, one after the other, and within 1 day, all these stages are spent, and on the second day of cleavage, a blastula is formed. It is a spherical, hollow, and ciliated structure, and it swims freely. Then gastrula. In the gastrula, the surface pushes into the interior at a point called the blastopore. ठीक है, एक बॉल है और फिर उसमें से invagination start हुआ. That means the surface pushes into the interior. आप ऐसे इमेजिन कर सकते हैं कि एक एयर फिल्ड बलून है अगर आपके पास, और उसमें आप फिंगर प्रेस करें तो एक होल की तरह से एक स्टार्टिंग पॉइंट हो जाएगा, एक एंट्री पॉइंट, और वो लेयर अंदर चली जाएगी उसके बलून के. सो इट इज दैट टाइप ऑफ स्ट्रक्चर. ठीक है, एक सेल की लेयर इनसाइड, एक आउटसाइड, एंड देन अ पोर फॉर्मेशन. नॉट अ हॉलो पोर, लेकिन कैसे, जैसे एक डिप्रेशन की तरह, लाइक इन इन्वेजिनेशन. सो हियर इन द gastrula, the surface pushes into the interior at a point. That point is called as blastopore to form a tube that will become the digestive system. And the blastopore gives rise to anus. That is why we call that a deuterostome condition. Deuterostome, embryonic development, and deuterostome, animal. So chordates and these echinoderms, they share this characteristic that they are having this deuterostome. Other condition is called as protostome, where the point at which this invagination or pushing in starts, that makes up mouth. In this case, that makes up anus, and mouth develops later in a different position. So the blastopore is the future anus of starfish. The cavity that is formed by invagination is called as archentron, or the primitive gut. In a gastrula, the cells on outer side form the ectoderm, and those lining the inner tube, that make up the endoderm, right? So now at this stage, it is a 2-layered early embryo. The cells migrate and multiply between these layers, and that will make up the mesoderm, the 3rd layer, right? So some cells might pinch off or develop from cell patches, and then they multiply there between these 2 layers, ectoderm and endoderm, and that will make up the 3rd layer, that is the mesoderm. So here in this picture, we again see this zygote, then this is a 4 cell stage showing in this, then 8 cell stage, and morula stage, which is, you see, that is a solid ball of cells, and the cells get progressively smaller in size. Then this division continues, and a blastula formation occurs, having a blastocele, and the cells somewhat are arranged on the periphery. Then this pushing in occurs here, the gastrula formation here. This part is the blastocele. I hope you can see this picture. It is visible to you. And then here, this archentron is formed. So this is the ectoderm and this is the endoderm, and between these two layers, the cells migrate and develop into the mesoderm. Then there is that, this structure is ciliated. This is the ciliated gastrula. The development continues, and further like more and more cells get differentiated and added, and this is the larval form. What type of larvae are formed, we will be discussing in some of the later slides. Now this is actual slide. Here you see the various types of developmental stages. For example, here you see 8-cell stage. Here you see blastula, the early blastula, late blastula. This is the late blastula, and then gastrula. Here you see that this archentron has formed, and this two-layered structure is formed, and that further develops. Here you see the gut is started forming here, and this is the larval stage, the early larva, and then the next stage larva, late larva, and then the juvenile that is developed, and from that juvenile that grows, and organs mature, and the adult mature, reproductively mature adults are formed. So during gastrulation, we were talking about these stages, blastula and gastrula. So we continue from that. During gastrulation, the advancing end of archentron bud off cells into blastocele and form the mesoderm. So this part, this is the advancing end here, right? This is the advancing end, this is the advancing end. So from here, the cells bud off, and they come to lie into the blastocele and form the mesoderm. Of course, this part, this space is blastocele here. This space is blastocele, and this is archentron. So some cells from this advancing end, they bud off and come to lie in the blastocele, and then they differentiate into mesoderm. Also, the advancing archentron form lateral pockets which pinch off and form the coelomic pouches. The coelomic pouches also develop from the advancing end of the this archentron. And these coelomic pouches later give rise to coelom. So these are coelomate, true coelomate organisms. And it's mesodermal lining and water vascular system, they are formed later. So coelomic pouches later give rise to the coelom, also its mesodermal lining, and the water vascular system. And embryo at this stage becomes a free-swimming larva. And since there is a larval stage involved, in case of C star, right now we are talking about C star. So this here, the development is called as indirect because it includes larval stages. Here, this is the thing we are talking about. So this is our late blastula, slight depression occurs here, and then more depression here, and blastopore formation, and this is the advancing part, and this is the archentron, right? And then from this leading end of archentron, here you see these coelomic pouches develop. Here, these coelomic pouches develop. So this is early larval stage, and the elementary canal develops more, ciliated band appear. हम उसके बारे में अभी आगे बात करेंगे. So this is a larval stage called as bipinnaria larva. Okay? Bipinnaria larva. So the late gastrula then forms this early bipinnaria, late bipinnaria. Here you see this is the side view of this late bipinnaria larva. And there is one more larval stage after that, that is brachyularia larva, which is having arms. And then that develops further, and then this juvenile C star is formed that grows in size. So we'll be talking about that now. So this is the early development where after zygote formation, after external fertilization, zygote formation occur, which is a 2N stage, and then the mitotic cell divisions continue. That is the cleavage part, and that leads to formation of morula. morula then transforms into blastula, blastula into gastrula, gastrula into the various larval stages and the adult stage. So now we'll be talking about these larval forms in echinoderms. So no other group of animals has such complicated metamorphosis in the course of development as these echinoderms have. Development may be direct, where there is no larval stage involved, or indirect, where various larval stages are there. In the direct one, the larval stages are missing. Indirect one, various life stages, larval stages, free swimming larval stages are there. Then in each class, now here we are referring to classes in the phylum Echinodermata. Of course, you can recall that there is Asteroidea, there is Echinoidea, Ophioidea. So there are several classes. You have to go through that classification to come to revise those classes. Holothuriidea is there, Crinoidea is there, Asteroidea, Ophioidea and Echinoidea. ठीक है, ये जो 5 क्लासेस हैं जिनके बारे में हमने बात करी थी इससे पहले कि कोई सी स्टार की तरह दिखता है, कोई कुकम्बर की तरह दिखता है, कोई सी लिली है, कोई सी अर्चिन है, बॉल की तरह है, सैंड डॉलर है, या स्नेक की तरह जिसके आर्म्स हैं, ठीक है, जिसको हम ब्रिटल स्टार भी कहते हैं. So in all these classes, there is a huge variety of larval forms. There are distinct larval forms. So they are, in each class, a few members are viviparous. That is, they brood their young in a sort of brood pouch on the surface of their body. So kind of they show viviparity. The development of larva takes place in a typical deuterostome fashion. In most cases, the characteristic free-swimming larva is developed externally, and these larvae are of phylogenetic significance, evolutionary significance. So now we come specifically to sea star first, then we will be talking about the larval stages present in other classes. Right now, we are talking about larval stages of class Asteroidea and specifically the sea star. So there are 3 larval stages, basically 2 larval stages, the Bipinnaria and Brachyolaria. But Bipinnaria can be divided into 2 conditions. One is early Bipinnaria, which closely resembles a hypothetical diplurula larva. It is believed that all modern echinoderms have been derived from a diplurula-like ancestor, right? So that stage appears in the early Bipinnarian condition. Then that leads to the typical well-developed, well-formed, mature Bipinnaria larva, leading to Brachyolaria larva. So you have to remember all these names, that which larval forms are there in which class. So here in the sea star, we have early Bipinnaria, which resembles diplurula larva, which was considered an ancestral, hypothetical ancestral, then a typical Bipinnaria larva and Brachyolaria larva. So let's first see how this development occurs, the life cycle. So here you see the egg and the sperms fuse, forming diploid zygote. This is called as fertilization, where gametes fuse and the diploid condition is achieved. Then mitosis, through several stages, through several cleavage divisions, makes this blastula. In the cross section, you see that cells are arranged in periphery, and there is a cavity there called as blastocele. That continues mitosis and changes in shape and elongates and forms gastrula. Now, in the case of gastrula, you see that cells invaginate. So there is an outer layer of cell and inner layer of cell. And then here we have this archenteron. And here you see these pouches have formed, which give rise to coelom in the due course. And then this develops here. This only one larval stage is shown in this. So this can be considered as post-embryonic condition, post-embryonic stage. And then the larva undergoes metamorphosis and becomes an adult. And then in adult matures, the gonads develop, and then they produce the haploid gametes, egg and sperms, and the life cycle continues all over again. Now, here are these larvae we are talking about. This is Bipinnaria larva, and this is Brachyolaria larva. Here you can clearly see that there are distinct arms formed on this structure. Here, in this structure, there are some lobes developed. This is early Bipinnaria. Then lobes develop, and then these lobes extend into arms. And then a small juvenile sea star is formed here. And then that matures into adult, grows into adult, and that form gamete, which are spawned, fertilization occurs, and the life cycle starts all over again. Now we want to talk about this stage, the bipinnaria, which comes after gastrulation. We talked about all these early embryonic stages in the previous slides. That is the morula stage, the blastula stage, the gastrula stage, and here then from the gastrula, this bipinnaria is formed. Now, how this bipinnaria, what are the structures in the these bipinnaria? So on the front side, there is a large preoral lobe forms and becomes bordered by preoral loop of cilia. ठीक है, क्योंकि ये एक्टिवली स्विमिंग है और सीलिया जो है वो फूड को प्रोक्योर करने में और फूड को डायरेक्ट करने में माउथ पे और फिर माउथ के अंदर फूड को पुश करने में these cilia help. So the ciliary arms are formed, arms having cilia are formed. So here in this case, at first the lobes are formed. The preoral lobe, and it is bordered by preoral loop of cilia. Then on each sides form the lateral lobes, which are bordered by postoral loops of cilia. These preoral and postoral loops arise by splitting the perioral band of cilia. So first a perioral band is formed, and then that divides into preoral and postoral. The larva is showing bilateral symmetry, that is left side and right side, they are same. So there is a left and right as well. If you remember, the adult one is having a radial symmetry, pentaradial in this case. This swims and feeds freely, and then it transforms into Brachyolaria larva. So first bipinnaria, then Brachyolaria. So this is a well-formed bipinnaria larva. Okay? This is just a diagrammatic figure hai, ye drawn figure hai. And here we see various lobes, जो कि अभी पूरी तरह से आर्म्स में डेवलप नहीं हुए हैं, ठीक है, आर्म्स ब्रैक्योलैरिया लार्वा में डेवलप होते हैं. दिस पर्टिकुलर फिगर इज ऑफ लेट बाइपिनेरिया लार्वा, where the lobes have produced, their lobes have elongated and has formed arms. So let's first see what are these labeling here. So this part, this is median, and it is present on the dorsal side. So that part will be called as dorsomedian arm. Then these are the lateral arms. Okay, shorter hai, these two. Number 2 is the lateral arms on the dorsal side. So dorsolateral. Okay, so this is dorsomedian. These are dorsolateral. Then posterodorsal arms at number 4. Here you see these number 4. 4 is side maybe, 4 is side maybe. Number 4 is the posterodorsal arm. Then we have at number 7 posterolateral arms here. These are the posterolateral arm, posterolateral arm. Okay? And then we have the other arms as well, venteromedian arm. Which number is that? Number 8. Let's locate the number 8 here. This is on the ventral side. So venteromedian, ये median है. And number 9, preoral arm, preoral on the ventral side. And then at number 10, we have postoral, right? So mouth must be located somewhere here, right? So mouth is located at 3 points. So mouth and preoral and mouth and postoral, right? So these preoral and postoral band, and we have then dorsal median, ventral median, and lateral, dorsolateral, ventrolateral, and so on. So depending on the location, they are named. If they are on the dorsal side, and if they are at the median location, dorsomedian. If they are dorsal side on the lateral side, that will be called as dorsolateral. If they are on dorsal and on the posterior side, so posterolateral. So that way, these arms or lobes are named. So this is a typical bipinnaria larva. Now this is the actual larva here. You see, these are several arms or lobes because they have not produced in those elongated arms as yet. So if you recall the gastrula is not having such type of extension. It is just an elongated, a barrel-shaped structure. But now the arms or lobes have developed on both dorsal and ventral side. And this is the gut that has formed, the larval gut that is developed here. This is the larval gut. Mouth is somewhere here, then leading to stomach, and here the anus. Here. So let's see in other one here. This is the picture that shows various lobes. Larva is usually planktotrophic, that is feeds by ciliary action, which beats, cilia beat plankton to mouth. They direct planktons to mouth, and then cilia are also, they help in locomotion. So cilia help in both locomotion and feeding. So here this is that bipinnaria larva. In this figure also you see early bipinnaria. This is early bipinnaria because the lobes are just forming in this, and on both dorsal and ventral side these are ciliary bands. Here ciliary bands are present. Here you see this is also an early stage where there are lobes and they are somewhat transparent, so you can see the gut developing inside. Here the gut is developing here, and these are flaps on the ventral side and on the dorsal side. So this is the same thing in various stages of development. So these dark margins show that these are ciliary bands. Ciliary bands are there here, postoral and preoral. This is postoral and this is preoral ciliary band. This is the one. Here. Then these lobes, they extend into arms and it transforms into brachyolaria. Now in this figure, this distinction between bipinnaria and brachyolaria is clear. You see, the dorsal side is this, the ventral side is this, and mouth is located here, right? And then these arms extend and make up this brachyolaria larva. I hope this picture is clear and you can understand how that structure develops from the gastrula stage. So let's come back to this part again, the development. So this was the gastrula stage here, archenteron formation, and then that continues. This is the lateral view showing further development of this and differentiation. Then early bipinnaria larva here, then the blastopore becomes anus, and the second opening here becomes mouth. So this is the blastopore. This is the original part from where invagination is started. And then this is the developing gut. The larval alimentary canal is further differentiated into stomach and small intestine. And here this mouth that leads through short esophagus into stomach and then into short intestine. And this is the exit part. So, and this is the ventral side and this is the dorsal side. So this late bipinnaria larva then develops these elongated arms here. And these arms, actually they don't have further use in this adult or young starfish development. They are shed off. They are important at first in swimming, and then they attach to some substratum. Suckers develop on these arms, they attach, and then they are broken off. And from this part, a young sea star develops, right? So this is the transformation from gastrula to the larva, to early larva, then late larva, then another larva, and then the young starfish. So let's continue with this. The uniform ciliation is there in the gastrula at first, and that is replaced by two ciliary bands. The perioral band around the mouth and the adoral band, which is there inside the mouth. perioral and adoral band. And then this perioral splits into two, that is preoral and postoral. So perioral band of cilia is around the mouth and adoral band of cilia is present inside the mouth. The larvae feed actively on the unicellular algae from water current, but cilia assist in that. Then this swim forward with the help of perioral band, and then it rotates clockwise and transforms into bipinnaria. So this text tells you about how gastrula transforms into early bipinnaria larva. Here you see, this is the bipinnaria larva, early bipinnaria larva. Still, as yet, here those lobes have not developed, which develop later. Now, the tip of the larval archenteron gives rise to two lateral pouches that develop into a U-shaped coelom, right? Then these pouches, they further develop, and the posterior end of pouches pinch off and form left and right somatocele. यानी कि फिर ये फर्दर डिफरेंशिएट हो रही है सीलोम into a exocele, into a hydrocele, and into a somatocele, right? So, you know, the purpose of all this is to achieve that adult stages and formation of various organs. First, the primordia of those organs, and then fully functional organs. So, from the coelom or body cavity, various parts of coeloms develop. That means the coelom differentiates into somatocele, and the somatocele is formed from posterior end of pouches, which pinch off and form a right and left somatocele. The remaining anterior portion forms hydrocele and exocele, which sometimes is considered together as exohydrocele. The larval gut is differentiated into mouth, esophagus, stomach, and intestine, as we have seen in some previous picture. Initially, there is just a primitive gut, which is called as archenteron. Then later, it is differentiated into mouth, esophagus, stomach, and intestine. The blastopore remains as larval anus. Then the right somatocele, and exohydrocele on the right side, they get reduced. Right? The further development occurs in the left exohydrocele because it is a bilaterally symmetrical larva. So there is, of course, there is a left part and the similar right part. So right somatocele and exohydrocele get reduced, whereas left exohydrocele give rise to water ring and radial canal. We know that there is this water ring, and then there is a central ring, and then there are radial canals. Exocele separates from the hydrocele and form the stone canal. If you remember, the stone canal is that canal that leads from that filter plate, that is madreporide, and leads to the ring canal. Okay? Ring canal में जाती है. So exocele separates from hydrocele and forms stone canal. The madreporite, which is the entry and exit point for water, that originates from the ectodermal invagination or right exohydrocele. So various structures, these embryonic structure give rise to various organs. Now this is the detail where you see this is somatocele. This side and on the next side, and between there is a stomach present. So around the stomach on both sides we see these somatocele, and then this is the hydrocele and exocele. So this is the coelom, which is differentiated into these various spaces, various pouches such as exocele, hydrocele, and the somatocele. And this part is giving rise to these ring canal and the, what do you call them? The radial canals. This is how from this part these structures develop. Then the larva, at the same time, when these changes are taking place and Bipinnaria larva is now having all these coelomes develop and water canal and ring canal and stone canal develop, we come to the next stage. That is, the larva transforms into Brachyolaria larva. So lobes of Bipinnaria develop into long, slender, ciliated, contractile structures called the larval arms. So from preoral lobe, if you recall, there was a preoral lobe, arise three short non-ciliated appendages. Short and non-ciliated appendages, relatively smaller arms, which are having suckers or adhesive discs at the ends. So there are these smaller arms from the preoral lobe, right? So these non-ciliated appendages or small arms develop, which have the adhesive discs. These are the fixing process. With the help of these, the larva, drifted larva, gets fixed to some substratum. Again, this larva is bilaterally symmetrical. It swims and feeds actively, and it gradually metamorphoses into a small sea star. This is the larva we are talking about, the Brachyolaria larva. Here you see there are these arms formed. Let's go through one by one what are these structures. So these are those small arms, okay, brachyolarial arms, which are having adhesive discs, which are non-ciliated. Then here, this is again the adhesive disc showing in these arms. Then three preoral arm. This is preoral arm. So from this part is developed. Then we have anterodorsal arm at number 4 here, anterodorsal on the dorsal side. Then we are talking about various structures here of intestine, right? 5 mouth के ही preoral or postoral हैं. Then we have small esophagus. Then we have this intestine, stomach, 7 part is stomach, intestine at this point, and then folded front, and that makes up the anus, the 8th point here. This is how this larva looks like. These dark points show the adhesive discs, and this is the gut, larval gut, and arm extensions. Here are these structures. Here you see these are the brachyolarial arm, the smaller arms, and then we have these elongated arms. These are further elongation. Here you see, and side by side at one point you see this pentaradial structure is forming. A primitive sea star is forming at this point here. Here you see, on one end a sea star is formed, and these are the arms. Then in the 6 to 7 weeks, Brachyolaria larva settles on some substratum. So all this development up to this stage takes about 7 weeks. Then this Brachyolaria settles on some substratum with the help of adhesive discs present on the fixing processes, representing a stalk on the anterior end. Then these later degenerate, larval mouth, anus, and ciliated band disappear, new mouth and anus formed, 5-arm rudiments appear around the oral-laboral axis here, as you have seen in the previous picture. Skeletal elements appear on the arms rudiment. We know that there is dermal skeleton with, in the form of ossicles. So at this stage, they start appearing, they start forming, they start developing. Radial canals then grow into these arms which have formed, the rudiments formed, and into this the radial canal grow. You see that they have formed from the exohydrocele at one point, and actually the hydrocele, the exocele, then later forming the stone canal. So then these radial canals develop into arms, then tube feet form, and then this particular rudimentary 5-armed structure that is detached from the stalk, and this is less than 1 mm with the short stubby arms. Now there are 5-armed structure formed, and then complex reorganizational changes result in the formation of adult sea star. Here you see. how somatocele develop, the right and left, and exohydrocele on the right side, that small, and on one side it develops further, makes up this ring, makes up these five extensions, and which then these five founding arms, these formative arms have formed, they grow into them and make the radial canals, and that the ring canal is formed at first, and then further structures are formed. And here, these adhesive brachiolarial arms, they are here. You see, they adhere, yeah, they are attached to some substratum. And this is how then the further development occurs and a five-armed structure is formed. Here you see in this one. So later, this breaks off, arms disappear, and this is released. This is small, juvenile, just a 1 millimeter small C star is released, and then arms then elongate, further development occur, growth occurs, and a C star is formed. So here you see in this, there is complete life cycle is given in this picture: adult male, adult female, shedding the sperms and the ova, and they meet. Millions of them are shed. Here, zygote formation occurs, fertilization occurs, entry of sperm and zygote formation occurs, fertilization membrane formation occurs to prevent further more sperms entering here. So that prevents polyspermy, then mitotic cell division, then morula formation, blastula formation, early gastrula, late gastrula. Here, let me enlarge it a little bit. And here you see the coelomic pouches formation occur. These pouches then give rise to somatocele, the exocele, the hydrocele, and then the early bipinnaria larva is formed here, still retaining this shape of late gastrula. Then the lobe formation start here, preoral, postoral, dorsal, ventral, and intestine, and development of this gut. And here you see the brachiolaria larva. And you see just at this point, the rudiment of a starfish formed. And these are the brachiolarial arm having adhesive discs through which it attaches to substratum. And when this primordial C star is formed, this breaks off, this part is released free, and then it develops. At first, the arms are small, then the arms elongate, the structures develop in them. This is the primitive water vascular system. Then they develop into that, achieve maturity, achieve reproductive maturity, and then gonadal development, and then formation of gametes and the release of gametes. So this is the life history, life cycle of C star. So with this, we end the class here. At this point, you know about two larval stages, that is bipinnaria larva and brachiolaria. On the next turn, we will be talking about other larval stages which are from different classes. तो ये एस्टेरॉइडिया के हमने लार्वा की बात करी है.